The purpose of this video is to review some details about the central dogma of biology. This will be a core concept not only in the lecture component of Chem 310, but as well as in the lab component, where we will essentially be studying the effects of an environmental toxin on each stage of the central dogma of biology. In lecture, we will provide a brief overview of the contents of a cell. So this would include general structure of the cell, as well as the general structure of some of the organelles and the function of some of the organelles within a cell. If you are still unfamiliar with this content or topics, please review sections in your textbook or sections from previous classes so that you have a full understanding of the purpose and function of organelles again and structures within a cell. So in this slide, we are going to cover step by step some of the components of the central dogma of biology, again with an emphasis on their role in the learning objectives for Chem 310. The first step or first component of the central dogma of biology takes place within the nucleus. So within the nucleus, hopefully everyone remembers that there are large molecules called deoxyribonucleic acids, and within certain stretches of these deoxyribonucleic acids, or DNA, are things called genes, right? So there are thousands, hundreds of thousands, if not millions, of nucleotides strung all together to form the genome within all the chromosomes of any given eukaryotic cell, right? And then again, within short stretches of those are components of DNA that code for genes, right? Genes that will eventually become proteins. So the first step that we're concerned with in the central dogma of biology is the conversion of DNA to RNA. So hopefully everyone remembers from genetics or again other classes in biology or chemistry that DNA is used as a template to code for complementary strands of RNA, right? So DNA is essentially read by an enzyme called RNA polymerase, which will read one segment of DNA or one strand of DNA and make a complementary RNA segment. And so initially this is copied, directly from DNA to RNA with corresponding nucleotides, right? So C to G, A to T, technically A to U because there's no thymine in RNA. But again, a strand is read, a DNA strand is read, and a complementary RNA strand is formed. Depending on what classes you had before, hopefully you remember that there's some editing done to that strand of RNA, but eventually it is shipped up and shaped out of the nucleus into the cytoplasm. And we're going to skip some of the details for this lecture, but this forms eventually messenger RNA. Right? And messenger RNA, again, is exported out of the nucleus into the cytoplasm, where eventually it is trafficked over to the organelle called a ribosome. And again, we're going to skim over some of the details here, but once the mRNA has reached the ribosome, it is essentially read in chunks of 3 nucleotides at a given time, right? And these are called codons. So each stretch of 3 RNA nucleotides are read and code for a corresponding amino acid. So in this chart up here, this is essentially the code that RNA codes for, and they're in 3 nucleotide sequences, and again, these are called codons, right? So you can use this chart to technically decode what a stretch of RNA nucleotides codes for amino acids, right? So in the top left here, we have U is an example in the top left. UUU codes for phenylalanine, right? Whereas down here, UUG codes for leucine. And again, you will have access to this chart, so if you're interested in reading or learning more about these, feel free to do so on your own time. I won't be requiring everyone to remember all the codes and codons, right? And so, again, skipping some of the details, these codons come in, they match up with something called an anticodon in the RNA, or in the ribosome, where essentially our DNA to RNA code is broken down into amino acids. These amino acids are then linked together to form a linear stretch of amino acids. This is called the primary structure of a peptide or a polypeptide. And again, this is just the linear linked together components of a protein, right? So amino acid 1, 2, 3, 4, 5, etc. These amino acids have just been covalently bonded together through the peptide bond. And as they come out of the ribosome, they begin in this linear sequence, right? Again, more details that we're going to skim over here. After this linear sequence of amino acids is strung together through components and properties of thermodynamics and many other things, our protein will eventually fold to form a functional, hopefully functional, three-dimensional shape, right? And down here in the GIF, in this GIF, we have a representative linear sequence of amino acids folding into some secondary and tertiary structures to form a functional protein. And we will revisit this GIF in lecture. But again, we start with this linear sequence of amino acids, and eventually we arrive at a three-dimensional functional peptide or polypeptide. So another step that can occur either before folding or after folding is a step called post-translational modification. If you break down the word, right? So this is post-translation. So after our RNA sequence has been translated to an amino acid sequence, post-translation, our amino acid sequence is modified, right? And here's an example of some of the post-translational modifications that can occur to a sequence of amino acids, again, whether that's before it begins folding or after it begins folding, right? So just some generic examples. Some methylation, where a methyl group is attached to our protein or amino acid sequence. Glycosylation, right, where a number of carbohydrates can be attached to our linear amino acids or our folded protein. Disulfide bonding, hydroxylation, ubiquitination, which is an important modification to make when you want to tag proteins to be broken down. And perhaps one that everyone's familiar with, phosphorylation, right? In some classes, phosphorylation is used as an example to activate or inactivate certain enzymes, right? So adding a phosphoryl group to a protein is an example of a post-translational modification that can inactivate or activate specific enzymes. So after our peptide or protein has been translated and modified, typically after that, our protein will, through other components that have been coded into our protein sequence, they will find their final functioning place within a cell, right? So whether that be up here in the membrane, so transmembrane proteins are coded, have special coding to help transport them there, or cytosolic proteins, right? And again, not to make this black and white, these are just two examples of locations that our final functional protein can arrive. So some of the steps we are going to analyze within Chem 310 is this step here, right? So what changes in a cell or changes in an organism can cause the transcription of DNA to RNA to increase or decrease? So the tool we're going to use for this is reverse transcriptase polymerase chain reaction. And then so over here, if we look at this, we are going to essentially analyze how much of a protein is expressed. So protein expression can go up, it can go down. Another component, let's use this as an example, we are going to evaluate how much functional enzyme is present within our samples, right? So through processes like protein expression, up and down, or post-translational modifications, most simplistically on and off, we can regulate how enzymes function, right? And that can be affected by being exposed to an environmental toxin. And then lastly, which I don't have an image for here, section 4, we are going to study the morphology or behavior of our zebrafish larva, which you can kind of think of as the end stage of changes, right? So we can change gene expression, and maybe that causes a malformation of some anatomical feature. We could change protein expression, which perhaps that decreases enzyme function, which then changes the behavior of a zebrafish, right? So those are the 4 sections. Section 1, essentially DNA to RNA expression. Section 2, quantity of protein expression. Section 3, enzyme assays or enzyme function. And then lastly, morphology and behavior. Now, you may be asking yourself, why do we care about things like DNA sequences and how much of a certain DNA segment is transcribed into RNA and how much RNA is transcribed into protein, or why we even care about all these tiny little nitty-gritty details in the first place, right? So, let's talk about that for a second. Over here, we have examples of 3 specific proteins from 3 organisms that are not humans, right? Not humans, not even mice. So we have green fluorescent protein, which can be found, or which was originally extracted from jellyfish. Luciferase, right? So the enzyme that's responsible for making lightning bugs or fireflies actually light up. And then these perhaps oddly placed light-sensitive ion channels that can be found in primal bacteria, right, or archaebacteria. And I've mentioned this before. The proteins, or the proteins from these little guys are tools that I actually used when completing my PhD, right? So maybe what you think of when people discuss genes is a linear kind of unilateral structure of DNA, right? Picture it as this individually colored arrow, and that you could start reading your DNA here and continue reading left to right in this example, and that's the DNA sequence for your protein of interest. And then down here, your corresponding amino acid sequence would be the same, right? Start from, say, left, go all the way to right. Perhaps in the past you've thought about genes or proteins, even for that matter, as just these kind of bland, unilateral, single things. One of the things we'll talk about in this class, and you will have the opportunity to research when we use our NCBI database accession for proteins of interest or genes of interest, is that in fact these, what you may have previously thought of as unilateral or bland sequences of DNA for a gene, are technically broken down into these things called motifs. For this purpose, let's discuss them as motifs rather than domains, right? So there are small sections of this single gene where there's a promoter section, right? So this helps promote the expression or translation or transcription of this gene. This stretch of DNA nucleotides codes for a sodium selective ion pore. Maybe this section of DNA codes for an enhancer element. Maybe this section codes for a trafficking motif, right? So a specific section of DNA that will help our protein of interest be deposited into the transmembrane space rather than in the cytosol. And then most genes have a stop motif, right? A common element of most genes is a section of DNA that will inevitably code for a stop codon to make sure that you don't just infinitely read RNA on forever and ever. Right? So if you break down your DNA into chunks, you can find elements like this, and you can even find common elements between different genes. So if we translate our DNA to RNA to amino acid sequences, we have similar segments of amino acids that essentially do the same thing, right? So this section of that linear amino acid sequence will code for perhaps an intracellular binding motif, right? So this is a section of the protein that's going to want to bind, let's call them things, right? Whether that's proteins or signaling molecules from the intracellular space. Here is that sodium selective ion pore. So we know that if you string these, this specific sequence of amino acids together, it will functionally form a three-dimensional structure that only allows sodium ions to pass from intracellular space to extracellular space. Right? Here's our traffic motif. So perhaps this is a section of your folded protein that needs to bind to another protein to allow it to get to where it has to go. Maybe over here is an example of phosphate binding motif. So this is a section of amino acids that will allow phosphate groups or phosphoryl groups to be added to a protein. And then maybe over here is our extracellular binding motif, right? A component of this protein that helps it arrive in the extracellular space where perhaps it can be activated by a ligand, or maybe it senses ion concentrations or something, right? So the notion that a section of DNA nucleotides is just this unilateral and unique, individual thing is technically incorrect. You can break that down into sections that can be used between similar proteins or different proteins, and each unique section of this DNA can have its own unique impact on that end functional protein. Now that we've discussed some of the finer details of information that can be hidden within DNA segments as well as amino acid segments, let's talk about how us as researchers, whether we're biochemical researchers or in this case perhaps even neuroscientists, why we care about what sequence of nucleotides or what sequence of amino acids codes for different things within a protein. Right? So in biomedical research, we're constantly developing new tools to help us answer questions that we frankly don't have the answers to. So I'm pulling information here from a paper by Gradinaru and Desaroth from 2010. This paper discusses the group's goal to try and build a light-activated chloride channel to be used to hyperpolarize or silence neurons, right? So they're trying to essentially build a protein that they can shine light onto, which will then selectively allow chloride ions to pass from the inside of the cell to the outside of the cell, or technically in this case from the outside of the cell into the inside of the cell, right? And they want to use this because this is a tool that can potentially turn neurons on and off, right? So ideally, what do we want protein expression to look like in this case? So I have this little image here in case anyone is unfamiliar with how the electrochemical mechanisms by which neurons talk to each other or send information from one end of a neuron to the other end of the neuron. So this is characterized by the action potential, right? So over here on our y-axis we have membrane potential. So basically this is the electrochemical state of the cell. Our resting potential over here, for example, is around negative 70 millivolts, right? And so when a neuron fires, the cell is held here by many proteins or enzymes, but specifically in this case, let's discuss the sodium potassium ATPase pump. So there's a pump that's constantly holding the electrochemical gradient of our neurons here at negative 70, right? So something will occur to this neuron and cause it to try and fire, which will, again, depending on the specifics, will cause the inside of our neuronal membrane to become slightly more positive, right? And at some point, there's a threshold of excitation or activation occurs, and voltage-gated ion channels, specifically sodium, will open up and sodium will flood into the cell, right? And so because sodium is positive, the inside of the cell will go from being, I say very hesitantly, but very negative to positive, right? So the electrochemical gradient of our neuron has changed. It's gone from very negative to very positive because those very positive sodium ions are flooding into the cell. Once your neuron has reached this point, another type of voltage sensitive ion channel opens, potassium in this case. So potassium sensitive ion channels open and potassium will flood out of the cell, right? So now we have a lot of positive things leaving the cell, causing our neuron to become very, very negative again, right? Positive things are leaving the cell, making it more negative on the inside of the cell. And this allows your cell to repolarize. We have this characteristic hyperpolarization wave here, and then after that sodium-potassium ATPase pump activity catches up to the changes that just perturb the cell, we return to our resting potential right around negative 70 millivolts. So that is a brief and not completely accurate summation of how neurons use electrochemical properties to communicate with each other, right? There's a lot of nuances that I'm excluding and a lot of details that I'm excluding just for the sake of conversation here. So we essentially want to introduce a protein into the membrane of a neuron that will allow us to exploit this type of wave here, right? So we want to put a chloride specific channel into the membrane of a neuron that we can shine light on that will allow chloride ions, again, which are negative, to flood into the cell, causing the cell to hyperpolarize, right? So we want to drive the electrochemical gradient of our neuron as far away from this threshold of excitation as possible. So we want to induce a big negative drop in our neurons, which will essentially silence them. And in order to do this, right, we want uniform expression throughout the membrane. In a perfect tool would have uniform deposition of our protein all over it, right? We don't technically want our chloride ion channel to only be expressed on certain portions of the neuron, and or we don't want it to not be expressed in the membrane at all, right? So if the job of this protein is to let chloride ions through the membrane, it doesn't do us any good if this protein is deposited somewhere within the neuron, right? So what this would look like if we were comparing our action potential over here is something like this. So up here we have our red trace, which is meant to be an example of what would happen if we had correct deposition of our chloride channel, right? We could shine light on it, which would cause our neuron to hyperpolarize. So here's your linear trace, and then this is what it would look like if we were monitoring that electrochemical gradient within a cell, right? Membrane potential in millivolts, negative 50, which is our threshold, and then negative 90. So every time we apply that light, we see a decrease in the electrochemical potential of our cell, right? So we're driving our cell more and more negative, making it harder and harder for that cell to fire. And then over here, this would be in the opposite situation, right? If we were trying to activate a cell, we could shine light. If we had a different protein that would allow us to turn neurons on, we could shine light on it and drive that electrochemical gradient closer and closer to that threshold to allow the neuron to fire. But again, we're trying to hyperpolarize or silence the neurons, so this is what we want our tool to look like. And so these are just examples of data from their paper. And if you choose to read this paper, it is relatively short, but the work that went into it is not short by any means. It takes a lot of time, thousands of dollars worth of research, and a lot of people to come together to exploit those simplistic components of our DNA sequencing and our amino acid sequences to allow us to put this into a cell and then use it. So on our previous slide, we had some bar graphs that quantified the success of this group of authors. But here, let's take a look at what our protein actually looks like, right, and the steps the authors took to achieving that success. So up here, we have a neuron, and we have our transgenic or artificial protein that we've taken out of archaeorhodopsin. So here we can just do it like this, right? So here's version 1. So version 1 is our protein that's been extracted from archaebacteria. So we have an archaeal halorhodopsin from a metazoan bacteria expressed in hippocampal neurons. So in this image here, we can see our hippocampal neurons, and our transgenic protein has been tagged with a fluorescent protein called YFP, right? And YFP stands for yellow fluorescent protein. So essentially, we've tagged our functional protein with a fluorescent protein that will let us see where our protein actually is. So in the first version, where we extracted the DNA sequence, which will code for the amino acid sequence, directly from a metazoan bacteria, and stuck it into the genome of our hippocampal neurons, and now our hippocampal neurons are literally just reading that DNA code, making the protein it codes for, and trying to express that protein, right? But doing this results in protein aggregates in the endoplasmic reticulum and intracellular accumulation, right? So this protein that's supposed to allow chloride ions to transport from outside of the cell to inside of the cell isn't actually getting to the membrane in sufficient quantities to do its job, right? We have intracellular accumulation, and technically the protein is aggregating in the endoplasmic reticulum. So it's not even being processed or transported correctly. So if we look over here to ENPHR 2.0, which, surprise, really just means version 2, our authors took this approach. So in version 2, they included or modified that DNA segment to include an endoplasmic reticulum export motif from a vertebrate inward rectifier potassium channel, which is, for our purposes, let's just say it's a special kind of potassium channel. But they took the endoplasmic reticulum export motif from this protein and edited the DNA segment and section and codes from our archaeal halorhodopsin, and now we have this partial archaeal halorhodopsin DNA segment, and then this partial vertebrate inward rectifier potassium channel DNA sequence, right? So by combining or mishmashing these two proteins, we've now prevented aggregate formation in the endoplasmic reticulum, but we're still not getting uniform distribution throughout the cell or expression in the cell membrane, again, as we need to, right? So here's our image. We have some better trafficking to axons or dendrites within our hippocampal neurons, but we still have a lot of intracellular accumulation. So if you've already looked over here to version 3, in version 3, our authors took a trafficking signal from that same inward rectifier potassium channel and stuck that trafficking signal or trafficking motif into our Franken gene. In doing so, we've now reduced intracellular labeling or intracellular accumulation, and we've improved labeling of cellular processes, right? So in version 3, now we can see a drastic reduction in intracellular accumulation compared 2 to 3 here, and we see excellent labeling of these dendrites or axons in our cultured hippocampal cells. So, and then if we zoom in, you can actually see, the columnar structure of this axon or dendrite here. So we see very, very good expression and deposition of our Franken gene or our chloride transport channel in the membrane of these hippocampal neurons, right? So this, in combination with the quantification bar graphs from the previous slide, demonstrate that our authors succeeded in developing a tool that will allow them to shine light onto these neurons and hyperpolarize these neurons, right? So now we have essentially a light-powered protein that can turn neurons off by combining genetic information from this arcyl halorhodopsin protein and combining it with an endoplasmic reticulum export motif from vertebrates, whichever vertebrate that is in this case, and trafficking signals from that same protein. So we've combined genetic information from multiple proteins to get this cool transgenic tool that will allow us to turn neurons on and off simply by shining light on them. Cool. And just to expand on the utility and diversity of transgenic tools that have undergone similar development as the example in the previous slide, let's talk about some of these proteins, right? So if you remember green fluorescent protein from one of our early slides, green fluorescent protein is actually, again, a protein extracted from jellyfish, and nowadays it's one of the most common cell labeling tools in the library of biochemists and cellular molecular biologists, right? That luciferase enzyme, again, the enzyme responsible for making lightning bugs or fireflies glow, has actually been taken out and used in several ways. So I have this GIF down here where the enzyme luciferase has been extracted from our lightning bugs and implanted into these mustard plants, right? And the authors of the paper that produced this GIF actually combined expression of the enzyme luciferase with a circadian rhythm gene, right? So basically a gene that its expression or activity changes with light cycles throughout the day. And they implanted it into these, again, mustard plants. So as light changes throughout the day, you see an increase in luciferase activity in these plants. And I don't know off the top of my head the purpose or the goal of this experiment, but I think it's a nifty way to show that, you know, you can combine two completely unrelated genes or proteins within an organism and do some really cool things in research down the road, right? So again, they've essentially combined genes that respond to changes in light cycle with this light that literally just makes cells glow or light up. So maybe more in line with biomedical research. So here's an example where these authors have taken cancer cells and transfected them, right? So we basically altered the genome of these cancer cells to express luciferase. So in cancer research, you can use this as a tool to monitor tumor growth. So in this paper here, we can see a timeline up at the top, day 3, day 15, day 22, day 28, and day 36, post-cancer cell injection. So essentially, these mice are anesthetized and have a small, tiny amount of cancer cells injected into their flank here. Then these mice can be treated with a drug, oxyluciferin, that will react with the enzyme luciferase and essentially make these tumors glow, right? And so over the course of this tumor development, you can see that the intensity of the light given off by this tumor increases more and more, right? So that we can use that protein from fireflies to monitor cancer growth over time, right? And one way we can use this in a positive way is that once we've artificially inoculated mice with these cancer cells that express luciferase, we can use that signal from luciferase as basically an indirect measurement of, again, tumor growth. So we can use it to monitor how rapidly tumors grow or cancer grows, as an example up here. And we can even use it to monitor how well a cancer neutralizing compound works. So in the example here to the left, this would be pre-cancer drug or perhaps a control, so no cancer drug. And then over here on the right is an animal treated with our compound that's meant to kill cancer. So you can use this technique to monitor cancer death as well. And then so over here, we have an example of how we've used green fluorescent protein from jellyfish to label neurons. So special and different kinds of neurons express different proteins, and you can use the regulatory components of those genes within special neurons to selectively express that gene fluorescent protein. Another tool that's been developed and used to label neurons is shown right here. As I'm recording this, it's a little glitchy, but you can use green fluorescent protein to actually monitor the development of the nervous system of zebrafish, the nervous system of zebrafish. So, I think this is one of the cooler uses of fluorescent proteins. You can literally monitor the growth of these neurons. It's pretty exciting and pretty cool.